Welcome to the Deep Dive. Today we're tackling something absolutely fundamental, something that impacts the safety of every single pill or treatment you might take.
That's right.
How we understand and monitor drugs after they've been approved and are out, in the real world.
Yeah, it's not just about getting approval.
This isn't just about getting a drug onto the market. It's really about a safety journey that lasts its entire life.
It's a complex topic, definitely, but one that's essential if you want to truly understand drug safety beyond just the headlines. We're going to go deep into this, pulling from, well, a stack of sources, primarily key excerpts from a foundational pharmacology text.
GOODMAN and GILMAN's.
That's the one. And our mission for you today is to really master this subject, to dig into the nuances, the challenges, the systems involved, basically understanding them like the experts in this field do.
Not just scratching the surface.
Exactly. This isn't just a surface-level skim. We want you to come away feeling you've mastered the details.
Right, aiming for mastery. So let's unpack this. Why is this phase after approval so critically important? I mean, the FDA signed off. It passed clinical trials. What else is there? Seems like it should be safe, right?
Well, the simple truth is pre-marketing trials, as essential as they are, they just can't possibly tell us everything we need to know about a drug's safety profile. History is, well, it's littered with examples that showed us this the hard way.
You mean like the thalidomide tragedy? That's always the big one people mention.
Exactly. A devastating example. Used widely outside the U.S. in the late 50s, early 60s for morning sickness, it led to horrific birth defects in estimates are over 10,000 children globally.
Terrible.
And the U.S. largely avoided the worst of it, really thanks to the vigilance of one person, Dr. Frances Kelsey at the FDA. She had concerns about the safety data. The evidence just wasn't there for her.
So her holding the line saved countless families here.
It absolutely did. And that disaster, along with others before it, underscored a vital lesson. Ongoing surveillance after marketing isn't optional. It's a public health necessity. You have to keep watching.
So out of that really tough history, what are the core principles now? How do we think about drug safety post-approval today?
There are three big ideas, really. First, safety and efficacy aren't static. They're not set in stone at approval. They are constantly being reassessed throughout a drug's entire life cycle. Approval is just the beginning,
Okay, so it's an evolving picture.
Precisely. Second, post-marketing safety isn't only about discovering incredibly rare dramatic side effects, although that certainly happens. Crucially, it's also about recognizing an increased risk of common events that weren't initially linked to the drug.
Okay, so not just a weird new rash, but maybe more heart attacks.
Exactly. Think about rofecoxib, Vioxx. The issue wasn't some bizarre new syndrome. It was an increased risk of myocardial infarction heart attacks. Heart attacks are common, unfortunately, but linking that drug to a higher chance of one, that was a critical post-marketing discovery.
Got it. And the third principle?
The third principle is that even drugs with known serious side effects can sometimes remain on the market.
Really? Even with serious risks?
Yes, if effective strategies are in place to manage those risks, and crucially, if the benefits for specific patients clearly outweigh them, thalidomide is actually a potent example here again.
How so?
It's now used very effectively for certain cancers like multiple myeloma, but it's used under incredibly strict controls. REMS programs will discuss specifically to prevent any chance of fetal exposure. The benefit is high for those patients, and the risk, while severe, is managed.
That really shows the balancing act involved.
Yeah.
So our deep dive today is focused specifically on this post-marketing world, identifying and managing these risks, constantly reevaluating that risk-benefit ratio.
That's the core.
And we'll be looking mainly at the U.S. system, right? Recognizing things might be different elsewhere.
Yes, primarily the U.S. system, though the underlying principles are pretty global. And just to clarify, there are other things that impact drug safety, counterfeit drugs, overdose, using drugs off-label, important issues. But our focus today is squarely on this process of finding and managing adverse events that occur after a drug is properly marketed for its intended use.
Okay, clear.
Right. And to really understand the systems we have today, you kind of have to trace back how they evolved. Often it was public health crises that drove the changes.
Makes sense. Let's start back in 1938 then. The Food, Drug, and Cosmetic Act. What happened there?
That act was a direct result of the sulfanilamide elixir tragedy. Just awful. Over 100 people, many children, died because a company used diethylene glycol, basically antifreeze, as a solvent to make a liquid version of an antibiotic.
Unbelievable. How could that happen?
It highlighted a shocking loophole at the time. Manufacturers didn't actually have to test new drugs for toxicity before selling them. The 1938 Act changed that fundamentally. It mandated pre-market testing specifically to prove safety.
A huge step forward. But not the last one, as you mentioned thalidomide earlier. That led to the Kefauver-Harris Amendment in 1962.
Exactly. While the U.S. was largely spared the worst of the thalidomide birth defects thanks to Dr. Kelsey, the global disaster was the clear catalyst. This amendment added another crucial layer. Drugs now had to be proven effective, not just safe, through well-controlled clinical studies before they could be marketed.
So efficacy was added.
Efficacy was key. And significantly for our topic today, it also made the reporting of serious side effects mandatory for manufacturers.
Okay, so safety testing first, then efficacy proof and mandatory reporting. Then we jump forward quite a bit to 2007, the FDA Amendments Act, or FDAAA. What prompted that?
That came after another big reevaluation, this time spurred by the withdrawal of Rofaxib, Vioxx, which we mentioned.
Right, the heart attack risk.
Yes. Rofecoxib was a very widely used pain drug, and the realization it increased heart attack and stroke risk after millions had taken it really shook things up. It prompted a major reassessment of post-marketing safety systems and led directly to FDAA.
And what did FDAA do? Sounds like it was significant.
It was. It beefed up the FDA's power considerably. It mandated the creation of a large population-based surveillance system, what became the Sentinel Initiative. It also gave the agency clear authority to require label changes, force companies to conduct post-approval studies if safety questions arose, and implement specific risk evaluation and mitigation strategies, or AMIS, which are tailored safety programs.
More tools for the FDA.
Definitely. And it also increased transparency by requiring clinical trial sponsors to post trial information and results in a public database, clinicaltrials.gov.
So again, disaster-driving reform. This history really sets the stage. But let's zero in now on why we absolutely need this robust post-marketing system. It all boils down to what pre-marketing trials can't actually do right.
Precisely. That's the core issue. Pre-marketing trials are designed to see if a drug works and is reasonably safe for a select group of people over a fairly limited time. If it passes that test, that's great. It's a necessary step. But it absolutely does not mean its full safety profile is known. Not even close in many cases.
And the first massive limitation is just the sheer size of these trials. They're small.
They really are, surprisingly small sometimes. Drug approval is often based on studies with just hundreds or maybe low thousands of patients. Data from 2015 to 2017 showed the median number of patients in the pivotal trials, the main ones used for approval, was just 467.
Wow. Fewer than 500 people.
Fewer than 500 in the median case. Now think about that compared to the millions who might eventually take the drug. Our sources give some striking examples from the past. Terfenadine, an early antihistamine, studied in about 5,000 patients, but eventually reached 7.5 million users before issues arose. Fenfluramine, part of the fen-phen diet drug combo, studied in only 340 patients before 6.9 million used it.
That gap is just staggering. Between a few hundred or 1,000 in trials and potentially millions in the real world. What does that mean for finding side effects?
It means you simply cannot detect uncommon side effects reliably, or even pin down the exact frequency of the more common ones. Think about it statistically. Imagine an adverse event that happens in, say, 1 out of every 10,000 people taking the drug.
Okay, pretty rare.
Pretty rare. Even in a relatively large pre-marketing trial, maybe 8,000 patients total exposed, which is bigger than most, you'd need that drug to increase the risk by almost double compared to placebo, a relative risk of nearly 1.9, just to have a decent statistical chance of even detecting that increase.
So if the risk increase is smaller, or the event is even rarer.
It's almost guaranteed to be missed, utterly invisible in the pre-marketing data. The source gives the example of liver damage with the antidepressant nifazodone. It caused death or the need for a transplant in about 1 case per 250,000 to 300,000 patient years of use.
Far too rare for trials.
Way too rare. Trials are nowhere near that scale. And these small studies also make it nearly impossible to identify specific patient subgroups who might be at higher risk. For example, it took post-marketing experience to realize that patients of African ancestry have an increased risk of angioedema, a serious swelling reaction with ACE inhibitors. That didn't pop out of the initial trials.
And then there's our individual genetic makeup.
Yeah.
That adds another layer of complexity that trials often miss.
It absolutely does. Pre-marketing trials just don't enroll enough people to capture the diversity of less common genetic variants that can turn out to be really important risk factors.
Like what sort of variants?
Well, consider the TPM2 gene. If you happen to inherit two non-functional copies, you're homozygous, which is about 0.3% of people of European ancestry. You can't properly metabolize thiopurine drugs like azathioprine, used for autoimmune diseases and transplants. Giving a standard dose can lead to life-threatening bone marrow suppression.
Right.
Or think about the discovery, which took time, that severe, sometimes fatal hypersensitivity reactions to the HIV drug abacavir are strongly linked to a specific genetic marker, HLA-B5701. Testing for that marker is now standard before starting the drug.
So genetics plays a big role in individual risk.
A huge role. Even common variations matter, like how we metabolize codeine through the CYP2D6 enzyme. Some people are ultra-rapid metabolizers due to their genes, converting codeine to morphine very quickly, risking respiratory depression. Others are poor metabolizers, getting little pain relief. These critical genetic links often only become clear once millions of genetically diverse people are exposed post-marketing.
Okay, so small trials miss rare events and specific high-risk genetic groups. What about the duration of these trials? You mentioned that's limited too.
That's another big one. Pre-marketing trials are often quite short. The median duration for those pivotal trials approved between 2015 and 2017 was just 24 weeks.
Only 6 months.
About 6 months median duration. But patients might take these drugs for years, decades, sometimes their whole lives for chronic conditions.
So effects that only show up after long-term use are completely missed.
Precisely. Serious adverse effects that require prolonged exposure, or maybe have a long lag time between starting the drug and the event appearing, they just won't be seen in a 6-month trial. Think about bisphosphonates for osteoporosis. Rare but serious side effects like osteonecrosis of the jaw or atypical femur fractures typically appear only after years of continuous use, way beyond the initial trial period.
And this also means we don't really know about long-term effectiveness either.
That's also poorly defined by short trials, which is why clinical practice evolves over time. For instance, the whole concept of taking drug holidays from bisphosphonates wasn't based on the initial trial data. It emerged later from clinical experience and subsequent research trying to balance long-term benefits and risks.
Okay, so short duration is a problem. And you also mentioned that the populations studied in pre-marketing trials are often very selected, very specific.
They are. Trials deliberately try to recruit relatively homogeneous groups. It makes it easier to see if the drug has an effect, the efficacy signal, and it helps minimize immediate risks that might complicate the study.
So who gets excluded?
Often, it's children, pregnant women, the very elderly, say over 75, people with significant other health problems like kidney or liver disease, or those taking a lot of other medications that could interact. Basically, the messier patients often seen in real-world practice.
So you approved the drug based on this relatively clean, homogeneous patient group, but then it gets prescribed to potentially everyone out there.
That's exactly what happens. Information on how the drug actually works and what its risks are in these complex real-world patient populations. The elderly person with 5 comorbidities, the patient with moderate kidney disease, the person on 10 other meds. That crucial information only really emerges after marketing as those people start using the drug.
Can you give an example?
Sure. Statins for lowering cholesterol. They're incredibly well-studied drugs overall, but clear data showing their benefits specifically in the very elderly and also quantifying the slightly increased risk of developing new-onset diabetes, that evidence came out much later, long after they were widely used, from post-marketing studies and analyses.
And the rofecoxib example fits here too, right?
Yeah.
The mismatch between trial population and real-world use.
A perfect, if unfortunate, example. It was initially approved mainly for acute pain, based on studies in relatively select patient groups, often for short durations, but it quickly became widely prescribed off-label for chronic conditions like arthritis, meaning people took it for much longer periods, often at higher doses, and these were often older patients with more underlying cardiovascular risk. That real-world usage pattern exposed a different population to a much higher cumulative risk than what was studied pre-approval.
Okay, another major limitation you flagged is the reliance on surrogate endpoints in trials. What are those exactly?
This is a really important and sometimes tricky concept. Surrogate endpoints are biological markers or measurements, things like blood pressure levels, cholesterol levels, blood sugar, A1C for diabetes, viral load for HIV, maybe tumor shrinkage in cancer, that are thought to predict a real clinical outcome that matters to patients, like preventing a heart attack, stroke, death, or feeling better.
So they're used as stand-ins to make trials faster or smaller.
Exactly. Measuring blood pressure change is quicker and requires fewer patients than waiting to see how many people have heart attacks over several years. So surrogates are used to reduce trial size and duration, especially for getting drugs approved faster.
But the big question is, do they always accurately predict the real outcome?
And the answer is, unfortunately, no, not always. While some surrogates are pretty good predictors, like HIV viral load is a very strong predictor of disease progression, others can be misleading, sometimes even dangerously so.
Like the Kefauver-Harris trial.
The cardiac arrhythmia suppression trial, CXT, is the classic sobering cautionary tale. Antiarrhythmic drugs like flecainide and incanide were fantastic at reducing the surrogate endpoint they suppressed, abnormal heartbeats, called ventricular premature depolarizations after a heart attack. Looked great on the EKG monitor.
So they worked on the surrogate.
Perfectly. But the trial was designed to look at the real outcome, mortality, and it showed that patients taking these drugs actually had a higher risk of dying compared to placebo.
Wow. So the drug fixed the surrogate measure but harmed the patient. A powerful lesson.
The profoundly important lesson about the potential dangers of relying too heavily on surrogates. And our sources note that surrogate endpoints are used quite frequently, especially for drugs that go through the FDA's expedited approval pathways.
How frequently?
Data from 2015-2017 showed 59% of pivotal trials use surrogate endpoints as the primary measure for approval. And for drugs specifically in those expedited programs, it was even higher, 67%.
So for a majority of new drugs, especially fast-tracked ones, we don't actually know their effect on hard clinical outcomes like mortality or morbidity when they first hit the market.
That's often the case. We know they affect the surrogate, but the link to the real-world benefit or even potential harm might still be uncertain at the time of approval.
The diabetes drug rosiglitazone, Avandia, is another example of this surrogate pitfall, right?
Yes, another major one. Rosiglitazone was approved based on its ability to lower blood sugar levels, A1C, a surrogate marker for diabetes control. Years later, a large meta-analysis of available data suggested it actually increased the risk of heart attacks.
Which led to big changes.
It did. That controversy was a major factor pushing the FDA to change its requirements for new diabetes drugs. Now, they generally require large pre-marketing trials specifically designed to look at cardiovascular outcomes, not just relying on the surrogate of blood sugar control and hoping to sort out CV safety later in post-marketing studies.
So that's an improvement.
It is an improvement. But even with these larger pre-marketing outcomes trials, they still face the fundamental limitation we talked about earlier. They might still not be large enough or long enough to reliably detect smaller to moderate increases in the risk of common cardiovascular events. Remember, Table 8.2, detecting subtle risk increases, takes huge numbers.
Right. Okay, one final limitation of pre-marketing trials mentioned is that they seldom use active comparators. What does that mean?
It means they usually compare the new drug to a placebo, an inactive sugar pill, rather than comparing it head-to-head against an existing treatment that's already used for the same condition.
Why is that? Wouldn't it be more useful to know if the new drug is better than the old one?
It absolutely would be more useful for doctors and patients, but the requirement from the 1962 Kefauver-Harris amendments was for adequate and well-controlled investigations to prove efficacy. Over time, this was largely interpreted to mean that placebo-controlled trials were sufficient, mainly because they are statistically easier and require fewer patients, and are therefore cheaper and faster than active comparator trials. Proving a new drug is better than an existing effective drug requires much larger studies.
So how often are active comparators actually used?
Not that often in the pivotal trials for initial approval. Again, looking at the 2015-2017 data, only about 29% of pivotal trials used an active comparator. Most used placebo.
So the implication is pretty stark. When a new drug hits the market, doctors and patients often simply don't know how its safety or its effectiveness compares to the treatments they're already using.
Precisely. Is it better? Is it worse? Does it have different side effects? Is it safer? That lack of direct comparative effectiveness and safety data at the time of launch is a really significant gap left by the pre-marketing evaluation process.
Okay, so let's sum up this whole section on why we need post-marketing surveillance. We've got small trial sizes, short durations, highly selected patient populations, frequent reliance on surrogate endpoints that might not predict real outcomes, and a general lack of comparison to existing treatments. It paints a very clear picture.
It really does.
It makes perfect logical sense then that, as the source states, new serious adverse drug effects are commonly discovered after a drug is already on the market and being used widely.
It's not just common, it's almost the expectation that significant new safety information will emerge. One study looked at novel drugs and biologics approved between 2001 and 2010.
What did it find?
It found that 71 out of the 222 approved products, that's nearly a third, had a major post-marketing safety event identified later. Things like being withdrawn from the market entirely, getting a new boxed warning, which is the FDA's strongest warning, or having a major safety communication issued.
Almost 1 in 3 drugs having a major safety issue found after approval. That's significant.
It is, and just general safety-related label changes are even more frequent. They affect something like 70% of drugs eventually. These changes often add newly recognized risks to the warnings and precautions section, or sometimes elevate risks to the boxed warning level, and these changes frequently happen years after the drug has been widely used.
So this really brings us to the modern philosophy, right? Like what the source calls a life cycle approach to drug safety. It's not just about the launch.
Exactly. The old way of thinking that a drug is basically considered safe enough because it got approved, unless proven otherwise, that view is really outdated and frankly dangerous, given all the limitations of pre-marketing trials we just discussed.
So the new view is?
The modern view, the life cycle approach, recognizes that safety and efficacy information evolves over time. We continue to learn about a drug throughout its entire life on the market. In many ways, the critical experiment for understanding a drug's true effects in the real world only begins after approval, when millions of diverse patients start using it under varied conditions.
And this modern approach relies on specific systems and actions to manage that ongoing learning process.
Yes, it's built on several components working together. Having organized systems for reporting and monitoring potential problems, sometimes conducting formal controlled studies after approval to investigate signals, and then having regulatory efforts to translate that new knowledge into actual changes in clinical practice to improve safety.
Okay, let's break down those approaches to post-marketing surveillance. The most basic level, the foundation, seems to be case reports and spontaneous adverse event reporting systems.
Absolutely. This is where it often starts. The systems like the FDA's Adverse Event Reporting System, or FAERS, collect reports of suspected side effects. These come voluntarily from healthcare professionals like doctors and pharmacists, and also from patients or consumers themselves, usually through a portal called MedWatch. Manufacturers are also required to report events they become aware of.
And the goal here is early warning.
Exactly. They're designed to be an early warning system, a way to quickly spot potential problems or patterns when a drug starts being used by large numbers of diverse people in the real world, outside the controlled setting of a trial.
And have these systems actually identified major issues historically?
Oh, absolutely. Simple case reports from observant doctors were the very first signals that raised alarms about thalidomide causing birth defects, and also about the diet drugs fenfluramine and phentermine, fen-phen, causing serious heart valve problems. These weren't found in trials initially.
And FAERS itself is a massive database now.
It's huge. It receives over 2 million reports annually. And importantly, the data in a de-identified form is publicly accessible through an online dashboard. The FDA actively monitors this influx of reports, uses data mining techniques to look for signals, basically, unexpected patterns of events linked to a drug, and then can decide if further investigation or regulatory action is warranted.
Okay, sounds like a vital system. But the sources spend quite a bit of time really detailing the significant limitations of these spontaneous reporting systems. Why are they so problematic if you're trying to truly quantify risk or prove cause and effect?
This is incredibly important to understand because people often misunderstand what these reports can and can't tell us. To know the true rate or risk of a side effect, you fundamentally need 2 pieces of information: the number of people who experienced the event, that's the numerator, and the total number of people exposed to the drug, that's the denominator. Spontaneous reporting systems are deeply flawed on both counts.
Okay, let's start with the numerator, the number of events reported. What's the issue there?
The single biggest issue is massive underreporting. Studies consistently show that the vast majority of adverse drug events that actually occur are never reported to these systems. One review estimated the median underreporting rate across 12 different countries was 94%.
94% underreporting, so only 6% get reported.
That's the median estimate. It means for every report received, there might be 15 or 20 other events that happened but weren't reported. And reporting isn't random either. It can be heavily influenced by things like media attention on a particular drug or side effect, ongoing litigation, how long the drug has been on the market. Reporting often drops off over time. This makes it totally misleading to simply compare the raw number of reports for drug A versus drug B and conclude anything about their actual relative safety.
Okay, so the numerator is incomplete and biased. What about the denominator, the number of people who actually took the drug?
That's essentially unknown from the reporting system itself. FAERS doesn't track how many individual patients received a specific drug, at what dose, for how long, or even for what medical condition they were taking it. You might try to get rough estimates using drug sales data, but that's a very imperfect proxy. It tells you how many pills were sold, not how many patients used them, or who those patients were. Information on usage in specific subpopulations, like the elderly or those with kidney disease, just isn't available from these reports.
So you don't know the true number of events, and you don't know the true number of people exposed. That seems like a fundamental problem for calculating risk.
It makes calculating actual frequencies or incidence rates impossible. And beyond just the numbers, the quality of the information in the reports is often poor. They frequently lack the critical clinical details needed to properly assess causality, the relationship between the drug and the event.
Like what kind of details are missing?
Things like the specific dose the patient was taking, how long they'd been taking it, what other medications they were on concurrently, their underlying health conditions, other potential risk factors for the event. Our source highlights a sobering point from an analysis of FAERS death reports. For about a quarter of them, the only information provided was death. No other description of the event, no clinical context, and even when other events were listed, crucial information like the actual cause of death, the patient's past medical history, or even the date they started the drug was frequently missing. You just can't determine causality with that level of missing information.
And this lack of detail and inability to calculate rates makes it especially hard to spot those smaller risk increases for things that are already common in the population, right?
Exactly. Because you can't calculate true rates and you can't adjust for the underlying health status of the patients taking the drug, these systems are inherently poor at detecting small or even moderate increases in the risk of non-rare events. Think back to heart attacks or strokes. These are unfortunately common, especially in older patients or those with diabetes or hypertension who are often taking multiple medications. A drug might slightly increase that underlying risk, say, by 20 or 30%. But in any individual patient report, it wouldn't necessarily look unusual or out of place, so it's less likely to trigger a spontaneous report.
Even though that small increase could have a huge public health impact if millions are using the drug.
Precisely. A small relative risk increase, maybe a relative risk of 1.2 or 1.3 for myocardial infarction, applied across millions of users can translate into thousands of excess heart attacks. That's a massive public health impact. But it's much harder for spontaneous reporting to pick that up compared to, say, a very rare but dramatic and unusual event like drug-induced liver failure, which is much more likely to stand out and trigger a report, even if the total number of affected patients is much smaller.
So these systems have an inherent bias towards detecting rare, dramatic, unusual events rather than subtle increases in common, serious ones.
That's one of their core limitations and biases. And because of all these issues, underreporting, unknown denominator, poor data quality, reporting biases, sifting through millions of these reports to find the true, meaningful safety signals that warrant further investigation is incredibly challenging. Even with sophisticated statistical data mining techniques, those algorithms are still working with fundamentally flawed and incomplete data. They can generate hypotheses, but they can't overcome the inherent limitations.
So, given all these major limitations, what are case reports and spontaneous signals genuinely useful for? Where do they provide value?
They're absolutely essential for generating hypotheses. They are the first alert, the smoke detector. A signal from FAERS doesn't prove causation, but it tells you where you might need to look more closely using more rigorous methods. The source emphasizes that case reports and spontaneous signals are most valuable for suggesting causality under specific circumstances.
Sure.
When the adverse event itself is very rare in the general population, when there are few other known causes or risk factors for that event, when the event occurs relatively soon after starting the drug, a close temporal relationship, and when the drug appears to confer a very large increase in risk for that specific event.
Can you give an example of that?
The example given is cerevistatin, another statin that was withdrawn from the market due to causing rhabdomyolysis, severe muscle breakdown, which can be fatal. Rhabdo is rare, but cerevistatin seemed to cause it at a dramatically higher rate than other statins. Our sources show data indicating the rate of fatal rhabdo per million prescriptions for cerevistatin was over 3.16. Compare that to rates like 0.19 for simvastatin or 0.04 for pravastatin at the time.
Huge difference.
A massive difference. That huge relative risk for a rare but very serious event, often with a clear time course after starting the drug, is exactly the kind of signal that spontaneous reporting systems can pick up relatively quickly and inexpensively, leading to focused investigation and, in that case, withdrawal.
Okay, that clarifies their role. They're the indispensable early alarm bell, especially for rare and dramatic events, but they can't tell us how often something happens or definitively why. For that, you need controlled studies.
Precisely. Once a signal emerges, or even just for ongoing monitoring, you need more rigorous methods. These fall into two main categories conducted after marketing: observational studies, often using real-world data, and post-marketing clinical trials.
Let's start with the observational studies using real-world data. What does that involve?
Right. This means leveraging large databases of information that are already being collected as part of routine healthcare delivery. Think about insurance claims databases from large health plans or electronic health records, EHRs, from big hospital systems or integrated care networks, or government programs like Medicare and Medicaid.
What kind of information is in there?
These databases typically contain information on millions of people, including what drugs they've been prescribed or dispensed, medical diagnoses they've received, often as billing codes, procedures they've undergone, maybe some lab results. You can try to link drug exposure in this data to the occurrence of specific adverse events or health outcomes.
Sounds powerful. Is it easy to do?
While it's generally less burdensome and expensive than setting up a brand new clinical trial from scratch, analyzing this real-world data is still a complex, time-consuming, and often expensive process. You need to identify the right databases, get access, handle massive datasets, clean the data, extract the relevant information, check its quality, and then apply sophisticated statistical methods to try and account for biases.
The FDA's Sentinel system is a big effort in this space, right? How does that work?
It is. Sentinel is a major initiative. It's essentially a distributed data network. The FDA partners with numerous large health plans, insurance companies, and healthcare organizations, currently over 30 partners covering well over 100 million Americans.
And the data stays with the partners?
Yes, that's key for privacy. The patient-level data stays behind each partner's firewall. The FDA develops standardized programs or queries, sends them out to the partners run the analysis on their own data using a common data model, and then they send back the aggregated results without individual patient identifiers to a central coordinating center.
So it allows the FDA to query data on millions without creating one giant centralized database of everyone's health info.
Exactly. It addresses some privacy concerns, and it allows the FDA to relatively quickly conduct studies on things like how often a drug is being used, or to investigate safety signals that pop up in FAERS. For example, Sentinel was used to confirm the link between the blood pressure drug olmesartan and a rare intestinal problem called sprue-like enteropathy, and also to monitor risks like intussusception after rotavirus vaccination. Sometimes a well-done Sentinel study might provide enough evidence to address a safety concern without needing to mandate a formal, expensive post-marketing clinical trial.
Okay, so what are the big picture strengths of using this kind of real-world data for safety studies?
They offer several key advantages over pre-marketing trials. First, they include massive numbers of people, often millions, who are much more diverse and representative of the patients actually using the drug in routine clinical practice, including those often excluded from trials, like the elderly, people with multiple conditions. Second, you can often follow patients for much longer periods looking for long-term effects. Third, because of the large size, you have a better chance of studying uncommon side effects, the effects of long-term therapy or effects in specific high-risk subgroups. And finally, they are generally faster and less expensive to conduct than starting a new randomized trial.
That sounds like a pretty compelling list of advantages. But of course, there must be weaknesses too. What are the major downsides?
There are two absolutely fundamental weaknesses that you always have to keep in mind with observational studies using real-world data. The first is data quality, and the second, and arguably bigger one, is confounding.
Let's talk data quality first. Why is it an issue?
Because this data insurance claims, EHRs wasn't collected primarily for research purposes, it was collected for billing, for clinical care. So it's often messy, incomplete, or inaccurate for research needs. Information on the actual dose of a drug taken, patient adherence, did they actually take the pills, use of over-the-counter medications, or important lifestyle factors like smoking or weight might be missing entirely or recorded inconsistently. Billing codes used for diagnoses can vary in accuracy. Tracking patients accurately if they move between different healthcare systems or insurance plans can be difficult. Some databases lack rich clinical detail, while in EHRs, a lot of crucial information might be buried in unstructured free text notes written by doctors, which are very hard to access and analyze systematically.
Okay, so the data itself had limitations. But you said confounding is the even bigger issue. What is that exactly?
Confounding is the Achilles heel, the fundamental challenge of all observational research. It arises because, unlike in a randomized trial, people who choose to take or are prescribed a certain drug are often systematically different from people who don't take that drug, in ways that are also related to the health outcome you're studying. And these differences might not be fully measured or captured in the database.
So people aren't randomly assigned the drug. Their underlying health or behaviors influence both getting the drug and their outcome.
Exactly. Doctors don't prescribe drugs randomly. They prescribe them based on a patient's health status, risk factors, severity of illness, other conditions, etc. Patients also make choices. So if you simply compare the outcomes of people taking drug X to those not taking drug X, you don't know if any difference you see is due to the drug itself or due to those underlying differences between the groups that you couldn't fully account for. This is confounding by indication or confounding by severity.
The hormone replacement therapy, HRT story, is a classic cautionary example of confounding leading researchers down the wrong path, right?
It is the absolute textbook example mentioned in our source. For years, large observational studies consistently showed that women taking HRT after menopause had substantially lower rates of cardiovascular disease, heart attacks, strokes. It looked strongly protective.
So doctors prescribed it partly for heart protection.
Yes, that became part of the rationale. But then, the large, rigorous, randomized controlled trial, the Women's Health Initiative, WHI, was conducted, and the results were shocking at the time. The WHI found that HRT did not protect against heart disease and might even slightly increase the risk of stroke and blood clots, and it clearly increased the risk of breast cancer by about 24%.
So the observational studies were completely wrong about the heart benefits. Why?
The most widely accepted explanation is confounding. The women who chose to take HRT in those earlier observational studies were, on average, healthier to begin with. They tended to be more educated, have better access to health care, have healthier lifestyles, less smoking, more exercise. It was likely these underlying health differences, these unmeasured or poorly measured confounders, not the HRT itself, that accounted for the apparent protective effect seen in the observational data.
That's such a powerful illustration. Even with huge data sets covering millions of women, the observational nature led to the wrong conclusion about cause and effect. Association is definitely not causation.
Precisely. And the HRT story, along with others, is why regulatory agencies and researchers are, quite appropriately, very cautious about making strong causal claims based solely on observational data, no matter how large the database is. You always have to worry about residual confounding.
But despite these major limitations, real-world data studies still provide critical information, right? Especially for questions you can't easily answer with a trial.
Absolutely. They are still incredibly valuable. They provide essential information on drug safety in real-world settings that would be difficult, impossible, or even unethical to obtain through randomized trials. The source gives a good example. Identifying the increased risk of developing type 2 diabetes in children and adolescents treated with certain antipsychotic medications, particularly second-generation ones.
Why is that hard to study in a trial?
Well, think about the ethics and logistics. It would be very difficult to justify randomizing children, especially those without severe psychosis, to take these powerful medications for long periods just to study the risk of diabetes compared to no treatment or a different treatment. But a large observational study using real-world data was able to follow kids starting these medications and clearly demonstrated about a 3-fold increased risk of developing diabetes compared to those starting other psychiatric medications. Figure 8-2 in the source shows the probability curves. That finding, despite being observational, was critical for informing clinical practice and monitoring guidelines for kids on these drugs.
So, observational studies using real-world data. Powerful tools provide unique insights, but require very careful methodology and extremely cautious interpretation due to data quality issues and the fundamental risk of confounding.
That sums it up perfectly.
Okay, beyond observational studies, the other main category of controlled post-marketing studies is actual clinical trials conducted after the drug is on the market.
Yes. These are formal, prospective, randomized controlled trials, RCTs, conducted after initial approval. They might be required by the FDA, perhaps to investigate a specific safety signal that emerged from FAERS or observational studies, or they might be conducted voluntarily by the manufacturer, maybe to get approval for a new indication or sometimes to try and demonstrate a safety advantage over a competitor.
And the goal here is rigorous assessment.
Right. Because they are randomized, they can, in theory, provide much stronger evidence about cause and effect regarding a suspected safety problem compared to observational studies. They can also sometimes uncover unexpected effects.
Like what happened with the obesity drug lorcaserin.
That's a prime example highlighted in the source. After several earlier weight loss drugs were withdrawn due to cardiovascular problems, the FDA required the makers of lorcaserin, Belvic, to conduct a large long-term post-marketing trial specifically to rule out excess cardiovascular risk.
And what did that trial find?
Ironically, the trial, called Camellia-TIMI 61, actually met its primary cardiovascular safety endpoint. It didn't seem to increase heart attacks or strokes. However, during the trial, an unexpected imbalance emerged. Patients randomized to lorcaserin had a slightly higher rate of developing various types of cancer compared to those on placebo.
Wow, completely unexpected.
Completely unexpected, and that finding, even though cancer wasn't the original focus, was concerning enough that it led to the voluntary withdrawal of lorcaserin from the market in 2020. It shows how post-marketing trials can sometimes reveal risks you weren't even looking for.
That raises a really difficult ethical question, though. If a safety signal emerges for a drug that's already on the market, and there are other, potentially safer alternatives available for the same condition, is it actually ethical to conduct a randomized trial where you deliberately assign some patients to the potentially riskier drug just to confirm the risk?
This is a huge ethical minefield, and the source touches on it. Imagine you have two drugs, drug A and drug B, both approved and effective for condition X. Then some signals emerge suggesting drug A might carry a serious, possibly fatal risk that drug B doesn't seem to have. Is it ethical to then design a trial where you randomly assign patients to either drug A or drug B?
It feels problematic.
It is. The ethical principle underlying RCTs is clinical equipoise, meaning there should be genuine uncertainty within the expert medical community about which treatment is preferable. If a significant safety concern has already arisen for drug A, even if it's not definitively proven, many clinicians and patients might reasonably believe that drug B is the safer choice. In that situation, true equipoise may no longer exist. Few informed patients would likely agree to be randomized to potentially receive the drug with the suspected serious risk when a perceived safer alternative is available.
The FDA-required trial comparing rosiglitazone and pioglitazone faced exactly this kind of ethical scrutiny, didn't it?
It did, significantly. Remember, rosiglitazone had the suspected cardiovascular risk signal, while pioglitazone, another drug in the same class, did not appear to share that specific risk at the time. The FDA mandated a large head-to-head trial, the TIDE trial, to compare their cardiovascular outcomes. This decision was heavily debated, and an Institute of Medicine committee reviewed the ethical considerations.
What did they recommend?
They recommended that mandating such post-marketing safety trials should only be done under specific conditions. First, there should be a transparent evaluation showing that existing observational studies cannot provide the necessary information. Second, the trial must be essential to answer a critical public health question. And third, there are heightened obligations to ensure potential participants fully understand the potential risks and the state of existing knowledge, especially if there's already reason to prefer one treatment over the other.
This whole ethical difficulty seems to argue for doing comparative effectiveness and safety trials much earlier in a drug's life cycle, maybe even as part of the approval process or shortly after launch.
That's exactly the argument made in our source material. The longer you wait, the more information, even imperfect observational data or clinical experience accumulates about the relative risks and benefits of different treatments. As that happens, it becomes progressively harder, both practically and ethically, to justify randomizing patients in a formal head-to-head trial because equipoise diminishes. Doing comparative studies earlier, when uncertainty is highest, might be a better approach, though often more expensive upfront.
Okay, this is a really comprehensive overview of how we monitor drugs post-marketing. We've gone from the historical reasons to the fundamental limitations of pre-marketing trials, explored the complex world of surveillance methods, spontaneous reporting, observational studies, post-marketing trials, and discussed their various strengths, weaknesses, and ethical challenges. But just identifying a problem isn't the end of the story, is it? You need to actually do something about it to improve safety.
Exactly. Finding the signal or confirming the risk is just the first step. The next crucial and often very difficult step is translating that new knowledge into actual changes in the behavior of prescribers and patients. How do you effectively communicate the risk and encourage safer use? This translation piece is also fraught with challenges.
What are the main tools or approaches regulators like the FDA use to try and influence that behavior and manage risks once they're identified?
The source highlights three common regulatory approaches. The first is communication trying to inform doctors and patients. The second is implementing specific risk evaluation and mitigation strategies, or REMS. And the third, most drastic action, is withdrawing the drug from the market altogether.
Let's start with communication. That seems like the most straightforward approach. Just tell people the newly identified risk. How is that done?
It seems straightforward, but its effectiveness is often debated. Regulators use a variety of communication tools. They might issue early alerts about potential signals emerging from FAERS on the FDA website, often in quarterly reports. The most common action is changing the official drug label, the prescribing information. This can range from adding information to the warnings and precautions section, or, for the most serious risks, adding a prominent boxed warning, often called a black box warning, at the very top of the label, highlighting potentially fatal or life-threatening risks.
Are label changes enough on their own?
Evidence suggests that simply changing the label, while necessary, may have a limited impact on prescribing behavior by itself. So the FDA also uses other methods like issuing formal drug safety communications, DSCs. These are announcements posted on their website, disseminated through email lists, sometimes podcasts, press releases, social media, and shared with medical professional societies, trying to actively push the information out. For really immediate dangers like contamination issues, they might issue urgent public health alerts.
So a multi-pronged communication effort. How effective is it really? Does getting the word out actually change practice?
Well, the evidence is pretty mixed and often suggests these communications are less effective than one might hope. Their impact seems to be quite heterogeneous and unpredictable. It's also really hard to isolate the effect of a specific communication because usually other factors are changing at the same time, maybe media reports, scientific publications, etc.
What do studies generally show?
Some key findings summarized in the source are recommendations for specific patient monitoring, like getting regular lab tests, often don't lead to sustained changes in practice. Warnings tend to have a bigger impact on decisions about starting new patients on a drug, rather than changing management for patients already taking it. Communications seem to be most effective when the message is very specific, when it's repeated over time through multiple channels, and importantly, when clear and readily available alternative treatments exist.
And you mentioned earlier, sometimes these communications can even have unintended negative consequences.
That's a really important point. Warnings, even when targeted, can sometimes have broader spillover effects. The source gives two key examples. FDA warnings about a potential increased risk of suicidal thinking with antidepressants in children and adolescents, and warnings about increased mortality risk when using atypical antipsychotics to treat behavioral symptoms in elderly patients with dementia.
What happened in those cases?
In both instances, the warnings led to decreased prescribing of those drug classes, which was the intent for the specific high-risk situations. However, studies found that prescribing also decreased in other patient groups or for other indications where the drugs might have been appropriate and beneficial. For example, antidepressant prescribing went down even for adults, and antipsychotic use decreased across the board in dementia, potentially leaving some patients' symptoms untreated. So, these broad communications can sometimes lead to undertreatment or shifts in prescribing with unknown consequences for patient health. It highlights the difficulty of crafting targeted risk messages.
Okay, so communication is necessary, but complex and not always effective. What about the second tool? Risk Evaluation and Mitigation Strategies, or REMS. What are those?
REMS are specific, mandatory risk management plans that the FDA can require for certain drugs with known serious safety concerns. The goal is to ensure that the benefits of using the drug outweigh its risks. A REMS program goes beyond just standard labeling and communication. It involves specific required actions.
What kinds of actions can be part of a REMS?
A REMS program can require one or more components depending on the drug and the risk. Examples include requiring that special medication guides or patient package inserts explaining the risks be dispensed with every prescription, sometimes establishing patient registries to track outcomes, requiring evidence that patients meet certain safe use conditions before they can receive the drug, like having a negative pregnancy test, mandating specific laboratory testing before or during treatment, requiring specific communication plans to healthcare providers, sometimes even requiring that providers undergo special training or certification to prescribe the drug, or that pharmacies be certified to dispense it.
That sounds potentially very restrictive. Can you give an example of a drug with a complex REMS?
The classic example often cited is isotretinoin, formerly Accutane, a highly effective drug for severe acne, but also a drug that causes severe birth defects if taken during pregnancy. Its REMS program, called IQLEDGE, is very complex and involves most of those components. Mandatory patient registration, provider certification, pharmacy certification, monthly pregnancy tests, counseling about contraception, etc., all designed to absolutely minimize the chance of fetal exposure.
Are these REMS programs actually effective in managing risk?
Well, currently, around 60 drugs have REMS programs in place. They are generally implemented based on sound reasoning about the specific risk needing mitigation. However, the source material notes that there has been relatively limited formal evaluation of how effective these complex programs actually are in achieving their safety goals in real-world practice. We assume they help, but the data proving their impact is often lacking.
Okay, and then the third and most drastic regulatory action is simply withdrawal of the drug from the market.
Yes, this is the regulatory equivalent of the death penalty for a drug. It's a very serious action and usually a difficult decision for regulators. They have to weigh the severity and frequency of the risk, the potential to manage that risk through REMS or other measures, the benefits the drug provides, especially if it treats a serious condition with few alternatives, and the availability of other therapies.
When does withdrawal typically happen?
It often happens when a major, previously unrecognized risk emerges after marketing, and that risk is deemed unacceptable or unmanageable relative to the drug's benefits. In many ways, a withdrawal represents a failure of the system to identify or adequately manage a major risk earlier in the drug's life cycle, either pre- or post-approval.
In these withdrawals, while obviously significant for the drug itself, can have broader impacts on future drug development and regulation.
Absolutely. They often have profound, long-lasting effects. When a drug or class of drugs is withdrawn for a specific type of toxicity, it almost invariably leads to heightened scrutiny and stricter requirements for evaluating that specific risk in all future drugs being developed, especially those in the same class or with similar mechanisms.
Can you give examples of that?
Sure. The source mentions how the withdrawal of several drugs back in the late 90s and early 2000s, like the antihistamine terfenadine, seldane, estemazole, the antibiotic grapafloxacin, the GI drug cisapride, all due to causing a dangerous heart rhythm problem called QT interval prolongation, led directly to the FDA implementing much more rigorous requirements for all new drugs to undergo thorough QT testing before approval. That's now a standard part of drug development.
And the anti-obesity drug withdrawals?
Same story there. The withdrawals of fenfluramine and dexfenfluramine due to heart valve damage, and later sibutramine due to increased risk of heart attack and stroke, led directly to the FDA imposing much stricter requirements for demonstrating cardiovascular safety, often through large outcomes trials, for any new anti-obesity drug seeking approval. These safety failures fundamentally reshape the regulatory landscape for future products.
Okay, we have really covered a lot of ground. We've traced the history and the laws, dissected why pre-marketing trials are inherently limited, explored the toolbox of post-marketing surveillance methods, spontaneous reports, observational studies, RCTs, and their significant challenges, discussed the actions regulators take from communication to REMS to withdrawal, and the difficulties in making those effective. Let's try to synthesize this and look towards the challenges and the future of post-marketing drug safety.
Sounds good. It's important to remember that pharmacovigilance, this whole field of drug safety monitoring, faces similar challenges globally, not just in the U.S. So international cooperation and harmonization of approaches are also really important. Looking ahead, several key areas stand out as drivers for future improvement and also present ongoing challenges.
Better access to high-quality data seems absolutely fundamental based on our discussion.
It really is a huge bottleneck and a major priority. Currently, access to large, comprehensive data sets, especially linkable data like Medicare claims combined with clinical information, is often restricted, expensive, and involves onerous approval processes, particularly for independent academic researchers. While systems like the FDA Sentinel have significantly improved the agency's ability to conduct timely studies, access for the broader research community often still relies on collaborations or specific FDA requests.
So what's needed?
There's a real pressing need for the development of large-scale, comprehensive, well-validated public data resources. Perhaps modeled on initiatives like the UK Biobank, these would ideally link different types of health information, maybe genomics, EHR data, pharmacy records, vital statistics while ensuring robust privacy protections, and critically, providing easier and less costly access for qualified researchers. Commercial databases are also emerging in this space, but having robust public data infrastructure is crucial for independent research and validation.
And with the potential for even more data becoming available, what about new approaches to actually detecting and refining safety signals within that data?
This is both a huge opportunity and a significant challenge. We're talking about potentially analyzing millions upon millions of adverse event reports, combined with electronic health records and prescription data for hundreds of millions of people. The sheer scale is immense. There's definitely promise in applying advanced computational techniques, things like artificial intelligence, AI, machine learning, and natural language processing to try and sift through this data more effectively to identify potential safety signals earlier or more accurately.
So AI could potentially help find those subtle patterns, those needles in the data haystack.
That's certainly the hope. And there's active research in this area, and this is a really critical, but highlighted in our sources, we have to be cautious. Even the most sophisticated algorithms are still analyzing data that suffers from the same fundamental limitations we discussed earlier, particularly the limitations of observational data.
Meaning confounding is still the issue.
Exactly. AI algorithms can be very good at finding complex associations and patterns in data, but they cannot magically eliminate confounding or overcome inherent biases in how the data was collected. An association identified by AI in observational data is still just an association. It doesn't automatically prove cause and effect. So while AI might help generate hypotheses more efficiently, those hypotheses still need rigorous evaluation using appropriate methods, keeping the limitations of the underlying data firmly in mind. We shouldn't expect AI to be a silver bullet that solves the core challenges of observational pharmacovigilance.
That's a really important dose of realism. What about using this growing data infrastructure for more efficient clinical trials? You mentioned pragmatic trials earlier.
Yes, there's a lot of interest in conducting large, simple, perhaps more pragmatic post-marketing trials that leverage real-world data infrastructure. The idea is you could potentially enroll thousands, even tens of thousands of patients remotely, maybe through patient portals or clinics, randomize them to different treatments, and then follow their outcomes primarily using routinely collected data from EHRs or claims databases or linked vital records. This could potentially make large safety or comparative effectiveness trials much less expensive and burdensome than traditional RCTs.
But these pragmatic trials have their own limitations too, right? They're not suitable for every question.
Definitely not. The source points out that pragmatic trial designs generally work best for studying relatively safe interventions that don't require complex consent processes, intensive safety monitoring during the trial, or endpoints that need detailed, structured clinical evaluation. The very features that make them pragmatic, often being open label, everyone knows who's getting what, having less control over other treatments patients receive, relying on real-world data for outcomes, can also affect the robustness and interpretability of the results.
How so?
For example, if adherence to the assigned study drug is poor in a pragmatic trial, or if patients and doctors frequently switch therapies based on their own judgment, which happens in the real world, it can muddy the waters and make it very difficult to interpret the results based on the initial randomization, the intention to treat analysis.
The adaptable trial, comparing 2 doses of aspirin, is given as an example of these challenges.
Yes, it's a great illustration. Adaptable was a large pragmatic trial comparing low-dose 81 milligram versus standard dose 325 milligram aspirin for preventing cardiovascular events in patients with known atherosclerosis, enrolling patients remotely. A key finding was that there was significant dose switching during the trial. Notably, 41.6% of patients initially assigned to the higher 325 milligram of dose ended up switching to the lower 81 milligram dose at some point.
So nearly half the high-dose group switched down. How did that affect the results?
Well, the primary intention to treat analysis, comparing the groups as originally randomized, found no significant difference between the 81 milligram and 325 milligram doses for the main cardiovascular outcomes. However, because of all the dose switching, the researchers also did sensitivity analyses trying to account for the dose patients actually took. Some of those analyses hinted that the primary outcome might have actually occurred more often in patients who predominantly took the 81 milligram dose. This leaves open the question: did the poor adherence to the higher dose potentially obscure a real benefit of 325 milligram aspirin? It perfectly illustrates how real-world complexities captured in pragmatic trials can make interpreting the findings challenging.
Fascinating. Okay, one last major area for the future highlighted is incorporating pharmacogenetics much more systematically into risk detection and management.
Yes, this is a rapidly evolving field with huge potential for improving drug safety. As we discussed earlier, our individual genetic makeup can significantly influence how we respond to drugs' both effectiveness and risk of side effects. Currently, around 300 drugs already have pharmacogenetic information included in their official FDA label. Some of these even carry boxed warnings related to genetic variants.
And this information relates to things like altered metabolism or specific hypersensitivity risks.
Exactly. It might relate to variants that increase the risk of a specific adverse drug reaction, like the HLA-B5701 link to a balk of your hypersensitivity we mentioned, or it might relate to genetic variants that affect how the drug is metabolized, like variations in the CYP2CC9 enzyme affecting warfarin dosing, or CYP2C19 affecting the activation of clopidogrel, Plavix. Understanding these links is crucial for personalized medicine.
And the importance of this is only going to grow.
Absolutely. As genetic testing becomes cheaper and more widespread, and as our understanding of gene-drug interactions expands, defining the clinical significance of these interactions and figuring out how to best incorporate this information into prescribing decisions, patient communication, and maybe even REMS programs will become increasingly important. We clearly won't be able to run large randomized trials for every single potential pharmacogenetic effect, so we'll need other robust ways to evaluate and act on this information.
The source mentions using genetic information itself as a way to predict drug effects through Mendelian randomization. Can you explain that again? It sounds powerful.
It's a really clever approach. Mendelian randomization uses the fact that common genetic variations are essentially randomly assigned to us at conception, like a natural randomized trial. If a specific genetic variant naturally mimics the biological effect of a drug, for example, a variant in the HMGCR gene that leads to slightly lower LDL cholesterol levels throughout life, similar to what a statin drug does pharmacologically, we can study the long-term health consequences associated with having that genetic variant in large populations.
So you're using the genetic luck of the draw as an unconfounded proxy for the drug's effect.
Precisely. Because your genes aren't influenced by lifestyle choices or prescribing decisions, studying the effects associated with these drug-mimicking genetic variants can give us clues about the potential long-term efficacy and potential side effects of targeting that biological pathway with a drug, free from the confounding that plagues traditional observational studies of people who choose to take the medication.
And this has actually worked in predicting side effects.
Yes. The source gives the striking example that Mendelian randomization studies looking at the consequences of genetically lower LDL cholesterol via HMGCR variants actually predicted the increased risk of developing type 2 diabetes that is now known to be associated with long-term statin use. That diabetes risk took decades of widespread clinical use and large trials to definitively confirm, but the signal was present in the genetic data much earlier. It suggests Mendelian randomization could be a powerful tool for anticipating certain long-term drug effects, both good and bad.
Wow. Okay, we have truly taken a deep dive into the world of post-marketing drug safety today. We've covered so much ground: the historical triggers and the landmark legislation that shaped the system, the fundamental, unavoidable limitations of pre-marketing trials that make ongoing surveillance absolutely essential, the different methods used for that surveillance, from spontaneous reporting with its critical flaws to controlled observational studies using real-world data and their challenges with confounding, to post-marketing clinical trials and their ethical complexities.
And then we looked at the actions regulators take to translate new safety knowledge into practice communication, REMS withdrawal, and the difficulties in making those interventions truly effective. Finally, we explore the ongoing challenges and future directions, including the need for better data access, the potential and pitfalls of new analytical tools like AI, the promise and limitations of pragmatic trials, and the growing importance of incorporating pharmacogenetics.
We really aim to go deep, as promised, covering the breadth of the source material, helping you, the listener, understand why simply approving a drug is just the first critical step, and why so much of the essential learning about its true safety profile happens only after it reaches the real world.
Right. And mastering these different layers, understanding the specific drug examples and what they teach us, grasping the statistical limitations of different study types, appreciating the ethical considerations involved in evaluation, recognizing the challenges of changing clinical behavior, and seeing the potential of future approaches like AI and pharmacogenetics. That comprehensive understanding is really what it means to grasp this complex, vital field like the experts do.
It certainly underscores just how much intricate work goes on behind the scenes, often imperfectly, constantly striving to ensure the safety of the medications that we all rely on at some point in our lives.
Indeed, it does. And perhaps it leaves us with a final thought for you to ponder, building on those future challenges we discussed. Given the ever-increasing complexity of new drugs, the diversity of patient factors, including genetics, and the sheer explosion of real-world health data, how can we best ensure that our future pharmacovigilance systems, even those potentially supercharged by AI, are designed in a way that truly learns from the hard-won, often painful lessons of past drug safety failures? Especially when these systems will inevitably still face the same fundamental limitations of observational data and the difficult ethical questions surrounding rigorous evaluation.
A really crucial question for the future of medicine. A lot to think about as you reflect on this deep dive. Thanks for joining us.